So it is my pleasure to welcome everyone to the first TCBG seminar of 2021. The year has started well, and hopefully we'll do the same with our seminars. Our first speaker is Dr. Arvind Ramanathan from Argonne National Lab. And before I introduce the speaker, I would like to just remind everyone we encourage that you turn off your microphones, but keep your videos on. So the video feed makes it actually more interactive, like a real seminar. And then usually we leave the questions to the end, but if you have a pressing question, you can actually maybe type it in the chat box. I will monitor those things and try to gently interrupt the speaker. Or if it's really, really pressing, feel free to jump in and ask your question at the proper time, but we want to minimize this so we don't interrupt the speaker's seminar as much as possible. With that, so let me welcome Dr. Ramanathan to the first seminar. By way of introduction, Arvind was trained as a computer scientist back home in India. And then he moved to Stony Brooks, where he did a Master's of Science, or MS, in computer science. So he's a computer scientist by background. And during his time there, he was also actually interacting with Brookhaven National Lab. Then for his PhD, he moves to the top university in computer science and computational sciences, Carnegie Mellon University, where he did his PhD in computational biology. And I think that's the period where he started applying computer science techniques to biological problems in general. Arvind then moves to Oak Ridge National Lab as a postdoc initially, but then he became a staff scientist there and a senior staff scientist there over years. At the same time, he also held a faculty position, a joint faculty position at University of Tennessee, until 2019, when he moved to Argonne National Lab, where he is now, and he's also a senior scientist at University of Chicago Consortium for Advanced Sciences and Engineering since 2019. So Arvind is, as my introduction suggests, sort of very interested in applying computer science and modern technologies and computational sciences to biological problems. Our last interaction, actually not myself, but my group, was on the work that they did together with the group of Romy Amaro, where they worked on coronavirus and spike protein, and how combination of simulation, application of AI and machine learning, which is the expertise area of Arvind, and I hope that we can hear more about that part of the work today. So that was our last interaction between Arvind and our group here. And I hope that after we learn more, that was why we invited you, Arvind, about your work and what you guys do to these systems, we might even expand our collaborations and interactions. Thank you very much again for accepting our invitation. We look forward to your seminar. Please.
Thank you. Thanks a lot. Thank you. Thank you for that wonderful introduction. And so, yeah, I'm going to talk about how we have been applying artificial intelligence targeted at COVID-19 drug discovery, but also by using that to drive multiscale simulations as part of this whole study. Of course, the study wouldn't have been possible without my collaborators, and I'm deeply indebted to them. I'll mention them explicitly as we are going through the slides. But this work is actually a consortium of over 300 people. So I cannot name all of them individually, but I'll definitely try and cover as many points as I can throughout the talk. Right? So come January or so, then last year, when the coronavirus epidemic started, Rick Stevens, who is the associate lab director for computing at our lab, decided that there needed to be a strategy to actually take forward in terms of how we could design drugs or find drugs that could be either repurposed or design new drugs based on existing ones for the coronavirus. And that was actually something that was the starting point of all of this work, in some sense, the genesis of this work, where I was already applying artificial intelligence and machine learning, or AI and ML techniques, um, with physics-based methods, but not towards actually drug discovery challenges, but towards more of the multi-scale ideas. And that's where Rami and I got together in late March or so, as we were doing some of the work, and then decided to combine forces because Rami comes with the expertise of modeling large systems. We wanted to see whether our AI methods could stand the test of large systems, and that's where we just decided to combine our efforts and do this. So in this talk, what I'm going to tell you is some of our experiences with using AI and ML by combining it with rigorous physics-based methods, and specifically for designing drugs. The second part is I also want to talk a little bit about what sort of challenges that AI and ML approaches impose on existing supercomputing platforms. And a last part of it where I'll talk about emerging heterogeneous platforms, especially in terms of AI hardware, and how we have been thinking of coupling them with MD simulations and so on. And of course, I'm going to talk about how we have been driving this towards, finding out small molecules that can bind to and inhibit SARS CoV-2 proteins, and some aspects of, the aspects of binding NSP1016 complex, if I have some time to cover that. I don't have to introduce the audience to the SARS-CoV-2 or the COVID-19 as such. This is actually a model that was drawn from Lorenzo and Rami. They were kind enough to actually share some of their initial models that they were doing. And so we started looking at the structure where you can see the virion structure is about 100 nanometers in diameter. It's a fairly large virus, unfortunately. With spike protein trimers that sit on the surface, expected to be about 100 copies per virion. The envelope is about 20 copies or 100 monomers that are present on the protein. The nucleoprotein, which is about 1,000 copies of which are estimated to be from the SARS-CoV-1. And the membrane protein, we expect about 2,000 copies of which are sitting there. And if you have many of these proteins, then the question becomes, how do you understand this organization of this virus? What are the things that it is doing? How can we design drugs? And obviously, a lot of people have been working on the spike protein. A lot of them have been looking at the internal structures and so on. But one of the advantages that we had with respect to the SARS-CoV-2 genome is the fact that Argonne was already pretty much deep in the depths of, understanding the proteins that are part of this virus. Especially the NSP structures, the non-structural protein structures that were solved in the lab through Andrei Yukhimiak, who is actually at Argonne and University of Chicago. A lot of these protein structures actually came from Argonne. So we had a rich source of structures that we could look at, including NSP15, NSP1016, PL protease, ADRP, MPro, and all these things that came out from these groups. And we could use them directly to start out for drug discovery processes. And of course, the part one of my talk right now is going to focus on, how did we target these molecules using AI, right? So this was part of this larger project from DOE, what we call as the National Virtual Biotechnology Lab. And molecular design actually focuses on two aspects, which is that we want to start out with these crystal structures that I just spoke to you about, and try and see how we can use physics-based and AI and ML models in group to assess the design fitness. What it essentially means is that we want to have some sort of desired activity across the protein. So this is the main protease, as you can see here, which is being targeted with a small ligand. And we wanted to end up with designs that actually look good with respect to the desired activity or desired biological effect, in which case you want to really inhibit the virus from doing what it is doing, and also having good physical and safety parameters. I'm not going to cover all these aspects today. I'm just going to cover the desired activity, which is to inhibit the protein of interest. So we'll be talking a lot of that. But the problem was actually to stand up such an infrastructure, you need to have a multi-stage computational pipeline that doesn't currently exist except for limited scales across different industries. Maybe some of the leading leading pharma industry actually has some parts of these infrastructure working. But the advanced computational infrastructure that supports high throughput screening, mode of action detection using MD simulations or other methods, as well as also design parameters using AI doesn't really exist. And so there was this need to actually put them up together in such a way that we could enhance the performance of these regular simulations by at least 100x, docking improvements at least by 50,000 times, which is impressive in some sense. But how to get there was actually the most important question. So the way people have been doing this, I don't have to outline really as to how much people have done on compound discovery, how do you identify things that are interesting, the toxicology aspects as well as the synthesis. But you have about 10 power 68 estimated drug-like compounds from which you need to really get to the point of getting a few that we can say are good hits against the virus. So in order to do that, this is where we started by looking at what sort of a pipeline would make the most sense to target the entire SARS CoV proteome as we know it. So this is actually a multi-stage pipeline where you can see there are three different stages that are involved. The first stage actually starts out by what we call as offline ensemble docking, which is the high throughput docking, which enables us to come up with lead molecules that might be good. But it's not important that you come up with lead molecules, but you also need molecules that actually have something which is related to the free energy of binding, or binding affinities are quite high, because throughput docking methods are usually limited by the fact that they give you a number, but these numbers are essentially, you can only trust them so much. So we needed to come up with some ways to actually impute or understand the binding affinity. And once we understood the binding affinity, we could go and ask questions about saying, how are these leads actually interacting with the different parts of the protein? What are they doing? And so if we find these stability or other features that the protein ligand interactions are important for, we could then input them into our machine learning codes that will actually enhance the performance of this. So as you can see, there's a fairly large set of people that were involved in this from the beginning, and at Rifan from your group, of course, Ahmad, has been kind of instrumental for many of the different stages as we run through them and so on. So the idea of building this entire workflow and taking them through the stages was actually kind of important for us. So the question can then be a very simple thing. Why not dock every available compound? So that's something that people like Virtual Flow have been doing, or if you've heard of it in the market, Google has actually invested a lot of this and so on. But the unfortunate problem is if you look at Autodock, or if you look at any of these different docking codes that are out there, assuming that you have all the 6 GPUs on a machine like Summit, which is our Oak Ridge leadership computing facility, and which is one of the top ranking compute engine on the world, you actually can screen only about 14,000 ligands or so per second. Right? That means if I take an entire library that has 1 billion compounds, it'll take me roughly about 8 hours to compute. This is great if you have one receptor molecule. That is, if you have just one crystal structure, or if you're doing one representative ensemble from an MD simulation. Now, just think of the fact that I just have 100 receptors to do. And if I were to run this, this is never going to finish running in some reasonable amount of time. I'll have to take about 72 days to get all of the receptors through my molecules, which is just not acceptable. So in that context, we said, okay, given that there is a lot of dearth in terms of this one, we needed to stand up a data pipeline that allowed us to have machine readable data set. Of course, a lot of the drug discovery community has been investing in it, right? I mean, the Zinc database, eMolecules, CureFFI, all these people have put out a lot of these molecules for docking programs. And your favorite docking program can actually do all of it. The unfortunate problem, though, is that they're not ready for machine learning, right? And as a context of which we had to put out this data in such a way that people could use it for machine learning. And that involves canonicalization, which includes, naming the subgroups in a formatted way, fingerprinting them so that you know what groups are present. You can actually search for them and so on. Also generate images that basically allows us to use like conventional algorithms on these data sets and put them all together. So this data infrastructure was something that we built in a very short time. In fact, within a couple of weeks of starting the project. And this resulted in about 60 terabytes of data getting released on the very first few weeks of starting of this project. So this is actually joint work with Ian Foster, Tom Breton, a whole lot of people from U Chicago also. But we had to use about 2 million compute hours to actually get there across all these computer sources, including Theta, Frontera, NVIDIA DGX systems, as well as the Argonne Leadership Computing Facility. But once you have that data, if I want to predict the docking score, if you think of how to predict the docking score, I can build a very simple regression formulation of the problem. So the given idea is you're given a protein structure, you're given a ligand, you want to dock it and you want to find or predict the docking score. I'll basically solve it using any form of regression that I like. It can be a linear regression formulation. I can have a support vector machine-like formulation. I can have all sorts of things. But the problem, though, is that we know that the docking results in general, especially the docking score, has some intrinsic biases. So Autodock has a bias for the docking pose. Some others have biases for hydrogen bond formation. Some of them are more electrostatically oriented. Some of them are like more shape oriented and so on. So in order to get rid of that bias, we said, instead of actually looking at the way to solve this regression problem, why not solve it as a ranking problem or a relative ranking problem? And that relative ranking problem is something that the machine learning community has tried to solve for a very long time. So the question then becomes like how you would think of movies, right? If I were to relate your, or if I ask you to rate the movies that you like, I can basically, on the basis of who your friends are or how you're related to the others, I can come up with a relative ranking for each one of your movies that you're interested in. So we applied a similar formulation. We said, okay, how many compounds can we find from the stock ranking list given some training data? And this still uses the regression problem, but instead of actually ranking it based on the bound conformations or something, we are saying how many of these compounds can end up becoming true hits. And that's actually the neat thing about it, which is that we can leverage image-based models, which are convolutional neural nets and things like that, which are really optimized for these large-scale data sets. If you have 4 billion compounds, it's quite a large data set. And now I can organize this into what we call as this regression enhancement surface plot. So we're going to talk about this protein called the SPL protease, which is one of the important drug targets for the COVID-19 virus. And we said, okay, for this particular protein, can you actually show me from the top Y percentage of ranked computed scores, how many of this predicted scores from my machine learning model would be true hits? That means if I were to stand at any point here, let's say I'm at 10 power minus 3, how many of the percentage of molecules should I have to see from my prediction scores to be confident that I would get all those top covered molecules? And it essentially means that I just need to cover about 0.36 or 36% of the molecules in order to make a detection that I have all the hits covered, which is actually good in the sense that you don't have to really dock a lot of molecules from this area, wherever you see the shaded ones in light blue. On the other hand, if I'm somewhere here, let's say I have the top 10 power minus 4 on one of these axes, and then that means that I need to screen at least 95, 98% of these compounds in order to be at least as confident in terms of saying, I have gotten the 2 hits of finding the docked molecule. So this allows us to reduce the space of screening. So we can actually get to a point where we can say, okay, you have so many compounds, let me apply only so many number of docking hits from my calculations and just run them through the screens. And that, if you actually incorporate it within the computational aspects of it, you can screen like the order of 6.25 million ligands against 32 receptors in a fairly short amount of time across Summit. What it also allows us to do is significantly enhance the utilization of the nodes. In fact, we can actually screen 317 to 10 power 6 ligands on Frontera, which is a TAC machine, Texas Advanced Supercomputer, on which we get pretty good performance in terms of how many docks that we can result in terms of per hour. And that actually led us to significantly enhance the scale at which we could dock, because now instead of doing using all the GPUs in terms of our numbers, we could actually get 319,000 ligands compared to the 14,000 ligands with just the high throughput docking. So this is a significant improvement in terms of the performance of how these runs could take place. And of course, this wouldn't have been possible without standing that workflow up, which was actually implemented using Ensemble toolkit, and I'll talk about that a little later too. So this is actually the part where we are using docking to drive this. But one other place where we thought that AI could have a significant impact was also to expand the chemical space in terms of how do we design new drugs for various molecules. So again, what you're seeing here is basically if you take any molecule, any drug-like molecule, there are a certain set of actions that are possible on it. So you can either add a fragment, remove a fragment, or you can do a fingerprint search and say what compounds are similar, or you can use a more sophisticated aspect like reaction synthesis model, which tells you how do we add and remove bonds and things like that. Of course, this is not being simulated, but we know that these are the actions that are possible on any given compound space. Now, what you could do is essentially use this existing structure and model all these different parts of the protein and so on. You can run some EnsembleMD simulations that allows you to gather some observables. This includes coordinates, the trajectories, potentially experimental data, maybe EC50s or IC50s, and as well as potential energy grids that tell you what sort of interactions are present between the ligand and the protein. Once that happens, we can also give it rewards values, which includes something like an MMGBSA or MMPBSA free energy estimate. These can be very rough. They don't have to be exact, but they can still be rough in the sense that you can get some values to relatively rank the compounds. And then you can get all these different metrics that you can use to say what sort of landscape are you sampling from. And then you can put ML-driven optimization or interface it with ML-driven optimization that tells you how to score these different molecules with respect to the different compounds and how they bind to and what they do. So this was something that we said, in order to actually look at it, we need to understand what that fragment search space looks like. And to go there, we said, okay, one way to think about it is to organize this entire thing into a graph. So if you think of this compound, let's say I take a look at this compound. This is formed of this phosphate in the middle, and then it has all these decorations hanging out of it. In my graph, what I'm going to do is basically take this node and represent that as a phosphate. And with each additional transfer I'm actually doing. So for example, if I move along the edge of each of this towards the leaf of this tree-like structure, you basically get this transformation that takes from this phosphate group all the way to each of the sub-additional groups that were added, and ultimately takes you to a leaf where it is the final compound. So this formulation, which allows you to modify this entire representation as a massive graph, allows us to think of ways in which you can use the docking score to navigate it to reach places which are completely like optimized for the protein of interest. So if you look at this part carefully, there are lots of things that are grayish in color. Those are nodes that don't lead you anywhere, which is in the sense that they don't have any binding for this particular protein, and so you can safely ignore them. But there are some parts of this tree which actually indicates that there are very nice lookout points, or basically what we call as starting points for design, where we can say, okay, this looks like a good candidate because it has the ability to bind to a protein because it has high docking scores. Again, high docking scores here means that I'm looking at the relative ranking that I was talking to you before. It's not about the absolute docking scores that I get from my simulations or virtual screen runs. So once you do that, you actually can use something like very simple page rank or random walk theory that allows you to dynamically prune this entire space and then only look for regions of the chemical spaces that are relevant for the binding across the target. So if I were to look at this problem, then it's about reconstructing a known set of inhibitors for a protein. So let's say I take an example of JAK2 kinase. It's a very famous example where we have a known drug like baricitinib, which is known to bind with very high affinity. So here is the final affinity of the scores, minus 15.93 using OpenEye docking package. And then what you can do is basically start out with the basic building blocks of this compound. So here are these basic building blocks. And then as I navigate through this entire tree, like what I was walking here, you end up getting better and better scores in terms of all the linkages that are going on. And not only are you doing that, you're also simultaneously improving the residue fingerprint, which is essentially to say, how are these different interactions that are working with the protein and how are they improving the overall binding score or not? And in some cases, we do fail. In some cases, you don't see that sort of binding happening. But in many cases, as we navigate this through this entire thing, we can reconstruct a top ranked compound across this library. Now, if I were to do this in an unsupervised fashion, I can use an algorithm like PageRank. This is very much like what Google does when you run, your favorite search term or whatnot. It finds the top ranked compounds across this one with a strong binding inhibitor for MPro. And what you see here are these 4 binding pockets, P1, P2, P3, P4. And we do find the compounds actually that have the binding to all the 4 pockets in the protein. Again, this is not by looking at anything other than the chemical organization and trying to expand the space and trying to say what sort of stable interactions are important and so on. I'm just using that information and coming up with a recommendation for compounds that have like good binding energy.
Can I ask you a question? I'm sure actually many of our molecular scientists students might have. So the one, I have 2 quite related questions. One is the chemical space that you define or the descriptors that you use for these many molecules. What are those? And then your objective function is the docking score that you...
Objective function, yes, is the docking score, which is based on the complementarity. Remember, I talked to you about the relative ranking of these compounds like this.
So you do docking for all of these, so you know the answer, and then that is your objective function. Now you use your machine learning to predict or guess that number as close as you can.
As close as you can, but with the relative ordering, which is important.
Ah, I see.
Relative ordering, which is important, right? If I were to do it as close as to the docking score, I can just use my regressor to basically do that, which is okay. I mean, you will get an answer, but they won't be true hits in some sense because you lose out on the resolution of what is relatively ordered in terms of what chemical spaces are useful or not.
I see.
That's actually the key thing here.
I see. And the descriptors you use for the molecule are just
yeah, the descriptors are quite flexible. So you can use as simple as an image. For example, here in this paper, we actually used images.
Oh.
Or you could use something like a chemical descriptor language that people have provided, like that includes the fingerprints or the moderate descriptors. There are many different descriptor things that we can use. In this case, we use the image because it was much more suitable, and most docking programs are good with these images, like SDF files and things like that.
Interesting.
But this is what we used eventually.
Thank you.
Yeah, thank you.
I have
That's
a
actually a great question. Please.
I have a related question. So my question is on the physics-based approach, the protocol that you explained about like finding, which incorporates ML as well as the descriptors and the MD simulations. So
Correct.
I am curious, like, does the prediction depend on the amount of simulations that you perform? Because what I assume is like the simulations that are being performed, we can because we have a big library, so we cannot go for extensive MD simulations. So most of the simulations might be trapped in kinetic traps, right?
Correct.
So does it if you iterate through this protocol, does it improve or does it remain the same?
I'll actually show you some results on that too. I've not come to the simulation part at all. Let me come there. I'll definitely address this question. Just hold on to your thought for a second. Right? Okay. So now what I can do is now with this very same idea, I can say, okay, let's start out with a known set of decoys. So I just showed you a construction of this known inhibitor, right? I did that just now. But let me show you a known set of decoys. And then what I want to end up doing is to get to a point where I can get it to something that looks like a known inhibitor. Okay, so in this plot, you need to be a little bit patient with me as I walk you over. What happens here is I have this molecule. This is the JAK2 kinase again, but with a decoy, that's a molecule that looks like a drug, but it is not an active inhibitor. And what I'm going to do is start out by running some simulations here, and I'm going to change these compounds as they run along, and then actually going to bound them with MMPBSA or GBSA calculation. In this case, it happens to be an MMGBSA. It's a rough estimate of the free energy of binding. And if you observe the left-hand screen, the compounds will change here. And the right-hand screen is actually tasting, or it's actually sampling and telling you what the GBSA estimates are. And then the idea is using this machine learning technique, I should be able to optimize to a point where I can get better binding molecules that at least look like a known inhibitor. And what you'll track here is the binding score for each of them that's entailed by each of these iterations. Okay, so at the top level, you'll see that. And if you look at what happens in iteration 1, iteration 2 is moving. Iteration 3 became bad. Then 4, 5, 6, 7, 8, 9, 10. And as that movie goes along, there are some small changes made to that molecule. Some of them might be reasonable, some of them might not be reasonable. But the general idea is that you do get molecules that end up reducing this GBSA score quite a bit as it is binding. Right? And in one case, it'll actually go up a little bit, and you'll see actually, made it very bad, and then comes back to the same norm. So what you're learning from this is that even within very reasonable number of iterations, within 25 iterations or so, we were able to take the dot score from about -5 or -6. You can see the peak here, which is about -5. It takes it all the way to about -15 or so, which is at least an order of improvement in terms of the docking energy. And in terms of the minima, we can actually see it as sampling a place where the GBSA score is actually quite low and nice in terms of binding for this molecule. And the fact that we can also sample conformational states that are relevant for binding, at least with 3 different binding pockets, 1, 2, and 3 that are highlighted here, all of which are close to each other that I showed in the previous plot, here where you can actually see the molecules moving around and doing all sorts of things. But it is also capturing the fact that there are some jiggling and wiggling that's going on with respect to the different side chains and whatnot, which allows us to capture these distributions and basically tell us what sort of things are going on. Of course, I've showed you here in terms of PC1 and PC2, which are the principal components, deducted from principal component analysis. We'll talk about the limitations of that a little later, but we are able to extract some of these properties, which are kind of important. So Shashank, this probably answers partly your question, but just wait for the little bit more of work. So what did that result in for, the SARS CoV-2, which is actually the main story, right? So our workflow does return better binding compounds. Here is where we scanned about 1,000 compounds. 50 of them show antiviral inhibition, which is good because you want to inhibit the virus as much as possible. A few of them actually, even without doing any simulations or anything, we ended up getting crystal structures. You can see one of the crystal structures here is the paper that we are currently working on with our collaborators at Oak Ridge National Lab, as well as Brookhaven. So you can see that it occupies P1 and P2, which is a small pocket, but it still has very stable interactions. You can see the difference between the predicted and the experimental structures. So the predicted one is shown in yellow, and the experimental structure is shown in cyan. Only the P2 pocket is a little different than P1, which is exactly the same. And now we are in the process of actually giving it to our synthetic chemistry collaborators so they can actually see what sort of differences are going on and how they can optimize this compound better. Right? So now that interesting question about what about sampling, what about getting stuck in kinetic wells and whatnot. And this is where we actually will target AI for helping us do multiscale molecular simulations. And if you look at the standard approach in which people have been doing simulations, is that you have a job scheduler on a big machine like Summit or Theta or any one of your favorite instruments. And so you run your job, you generate all of the simulation data, you store the data, you do the analysis, and then do the visualization. At the end of it, you can get a whole bunch of biologically interesting events that basically allow you to look at some events of interest. But when exascale machines are going to come up, this aspect of storing the data and analyzing it afterwards with hundreds of terabytes of data is just not going to be possible. So just to give you an idea of how much of our SARS CoV-2 simulations generated, we have approximately 45 terabytes of raw data that's already sitting. This is all processed data. And I think the raw data is even much higher than that. And if we even spend like 2-3 years analyzing the data, we wouldn't have the ability to go through all of that has been going on. You can ask Anda about the more details of 305 million atom system, what was simulated, what sort of things we are seeing from Rami's data and so on. But the idea here is that it would just be humanly impossible to spend time to looking at these biologically interesting events. At which point we said we need to really couple the simulations together in what we refer to as in situ analysis. And this results in reduced data movements, as well as providing online monitoring and feedback, which is actually the key thing that enabled us to do what we did for the NAMD paper, which was nominated and won the Gordon Bell Prize. So this uses the ensemble toolkit workflow, which allows you to essentially instantiate n number of simulations, collect all the data, run your training and inference at the same time. So you can actually do this enough number of times. And then based on some idea that you have interesting events that you detected, you start out with new simulations, and I'll detail that a little later. But one of the things that was actually a barrier for us was to see how do we use machine learning data or machine learning techniques on MD simulation data. And this goes back to some work that we started doing in 2015 and 16, where we were looking at the statistics of fluctuations of atomistic simulations. And one of the things that we observed immediately was that there were long tails in these fluctuations. And the long tails in the fluctuations give rise to what we call hierarchies in machine learning. It's a signature of most machine learning techniques, which is that if they see long tails, in imaging you see this, in videos you see this, in many natural settings you would see long tail distributions. Moment you see them, it means that there is a hierarchy of scale, which means that smaller scale fluctuations are coupled to larger scale fluctuations. And we wanted to basically find out ways to extract that. So there are linear ways to do that, which includes principal component analysis, which unfortunately does not pay any attention to the tails. On the other hand, deep methods or deep learning methods can actually pay attention to these tails and also take care of intrinsic non-linearities in the system. So we introduced this technique, what is called as convolutional variational autoencoders, where it ends up building a low-dimensional representation for states from simulation trajectories. People have done, time-independent component analysis, which is something that Frank Noe and others have done. There's principal component analysis that includes the temporal aspect. That's another story. And there are many different ways to actually reduce the dimensions. But we said, okay, since the data is essentially nonlinear, it has quite a bit of nonlinearity in the data, a technique like deep learning can actually learn from it. And we said, can we actually understand what it is doing? So if you build this convolutional variational autoencoder, it projects the data into this low dimensional manifold where you can see each dot represents a conformation from my simulation. And I've painted them with some sort of reaction coordinate. So in this case, it is a fraction of native contacts, what you can see. And there's a protein called as the FS peptide. It's a fairly toy example to understand. And if I were to stand at the origin, right, at 0, 0, 0, and move along each of these dimensions, what I essentially see is that I either move to a completely folded state, what you see in blue, or you move to a completely unfolded state, what you see in red, and there are all these intermediates that you cover. Again, more than 2 dimensions are hard to visualize, so you can actually use a method like TSNE or something, which allows you to visualize the space in a low-dimensional manifold. So this manifold is actually projected to 2D. And what you see from there is this very defined well here, state 1, which is the folded state. And from 2, 3, 4, 5, 6, 7, 8, 9, you can track the different conformational changes that are going on. And they all represent different intermediates that are there as part of this folding trajectory. And what we have seen from there is that basically, if you apply this method across multiple protein system and so on, it provides a general way to extract such collective coordinates or reaction coordinates, however you want to call them. Basically allows you to quantify how do you get from unfolded state all the way to a folded state, which is if I move in this direction, I can basically see the transition happening. Or each of these directions actually gives rise to a new way to sample the folded state in a nonlinear manifold. So we said, okay, why not do this as an adaptive ensemble technique? So this is actually a part where, in collaboration with Shantanu Jha at Rutgers and Brookhaven, we said, okay, can we stand this up in such a way that we can fold proteins? And so this is actually where you start with simulations across multiple GPUs. Obviously, ensemble simulations have their own power. You collect the data, you push it through the VAE models, choose the best VAE based on the hyperparameters that you're using, run this VAE to come up with inference on saying what constitutes a novel state versus not. If you come up with a new novel state, then you say, okay, I'm going to start new simulations from them. If it's not, I'm just going to cull those simulations because they're not essentially productive or anything. And you're going to iterate that until the training cycle of the protein is done folding or whatever that is of interest to you. And so there are two phases here. One is the learning phase where the MD simulations continue to run for a slightly longer time until the machine learning takes over. And then basically tells you whether it is able to do well or not. And of course, it's exploiting a part of this exascale software toolkit that we are building, what we call Candle. I won't talk about that here. It's not that relevant. But the idea was to use this infrastructure. So what does this buy us? From an AI-driven point of view, there are at least 2 orders of magnitude better in terms of traditional sampling. So what we have done here is we are comparing our trajectories with Antron trajectories, which is the reference or standard way to kind of say, okay, if we fold a protein, can we look at all the states that were sampled? So here you're looking at the percentage of explored population on the y-axis and the absolute simulation time on the x-axis. Here is VHP, which is a protein on which we did some folding experiments. And what we observe is that there is a crossover point at some point at around 1 microsecond or so, where the machine learning actually kicks over and starts sampling at a much faster rate compared to what the Antron trajectories can do. So here at about 22.8 microseconds, we actually cross quite a bit of what the Antron simulations have seen thus far. And this holds true with even BBA, again, with like half the simulation or just 1/4 of the simulation we're able to do. That's 1 order of magnitude impact. But let's say I have the ability to take the Antron trajectory, train my model, and then start simulations from some place. At which point you can actually see there is another order of magnitude speed up where I can actually, just by using about 200 nanoseconds of simulation, I've actually covered a lot of the space and then, start taking over in terms of, the sampling. So here, what we are seeing is compared to what weighted ensemble simulations do, which is something that Lillian Chong, one of my collaborators, is working on and so on. It's in a similar manner, except that this is actually using the deep learning all the way through from the beginning till the end in terms of saying, can we fold proteins? Can we actually look at ways in which we can accelerate these methods and so on? And this is something that we applied on the spike protein system. Of course, that gives rise to these MD simulations that's coming into the latent space and you're simulating it and then saying there are some challenges associated with it, which is that the representation of maps as sparse matrices can pose challenges on deep learning, as well as the fact that the parameters for training that you need for the spike protein, which is about 1 billion, is much harder to train on a single GPU. So we, of course, went through the process of implementing it across GPUs and whatnot. But unfortunately, that's not quite useful. So we said we needed to come up with a new way to think about it, at which point we said we are going to apply something called as the adversarial autoencoders, which allows us to directly work with what we call point cloud representations, basically the XYZ coordinates of the protein. And you push it through the same encoder decoder model that I was just talking to, like in the previous set. But this projection is now balanced out by a prior, which is based on all the training data that you might have had access to from the simulations. So I had this weighted ensemble data that Lillian gave us for the spike protein, which you could use to actually train the model. And it actually has much fewer parameters. It's only about 10,000 parameters or so, which allows us to train much faster at the scale of these simulations. So you can see the memory performance, whereas for the spike trimer, the memory consumption for the VAE is actually quadratic. On the VAE side, on the adversarial autoencoder side, it's more or less linear. And the time for training per epoch is also pretty much constant. So there's a linear increase in the use of the memory, but almost constant cost in training time, which allows us to better scale on very large systems. So if we end up doing that, we can actually learn a lot from our MD simulation. So what I'm going to show here is the projection of the spike data onto this low dimensional manifold that is spanned by this. And each of these dots is actually a confirmer from my simulation. And I need to thank John Stone for actually generating this amazing picture, along with Anda, who also did a significant portion of this visualization piece. So what you can see is each of the trajectories are now spanning a different portion of this landscape. And then what we see is we can actually understand what sort of conformational changes are going on with respect to this closed state. So here is actually the closed state of the protein. The RBD is actually closed. And then what you see again is the fact that we can also track some of these conformations. Oh, sorry. We can also track some of these conformations where you can see it is going from the completely closed state to an open state, and those are all mapped differently in the different parts of this landscape. Again, observe that this is a 3D representation of a much higher dimensional landscape, so it's very hard to kind of say where are these separate clusters forming and so on. But in this manifold, if you understand that they are all in a nonlinear manifold, you actually can map out the different states and how these differences are. And that's all you care about in terms of the difference between the open and closed states. Now, I can use this information to kind of map it back onto this spike ACE2 complex and ask what sort of conformational changes I observe. There's a very limited portion of the landscape that's actually relevant. And what you see here is one of those states that I'm going to pick up as an outlier from our analysis. And that outlier actually shows some very different orientations of the stalk region of the protein. I'm just showing the entire structure to keep it in perspective. But the idea is that one of these outliers does show significant conformational changes in the spike as a consequence of binding to ACE2. In a similar manner, you can also see some of the structural changes where you can do the different orientations and you look at different sort of changes. What does it ultimately bias? It's the fact that DeepDriveMD allows us to gain effective scientific performance where you can see with respect to this knee angle that I'm tracking with respect to the spike in the ACE2 complex, you can clearly see that with respect to one of the first set of MD simulations, which did not have any machine learning input to it, we can see that it's only sampling around 35 degrees or so. But as we improve that and we put it from the deep learning, we can actually shift that, this one quite a bit. We can actually shift the knee angle to about 40, 42 degrees, which is a significant acceleration where you can see without the DeepDriveMD about 0.5 microseconds of these simulations, we were not able to sample as many of these states, but with DeepDriveMD, within about 60 nanoseconds of this simulation, we can really push forward on terms of the sampling efficiency. Overall, we also observed 25% more conformations of the knee bending with only 12% of the time. In fact, Anda is in pretty much analyzing these simulations with Lorenzo and others at Rami's group in terms of understanding what sort of conformational changes are being enabled by this entire sort of sampling that we're doing. And in fact, with replicas 7 to 9 or so, we even push it forward. The knee angles are even more bent. And that's kind of interesting because we did not have that as an idea for selecting, or any of these things were not input as a reaction coordinate. This is something that the AI discovered, in the sense that the deep learning idea provides us with a mechanism to learn about it. And then we use that to shift what sort of conformations we are seeing from these simulations. And of course, we have been able to sample this to, scale this to about 1024 nodes on Summit. And now coming back to this docking story, as to what happens when we run these sorts of simulations, we first say we get a coarse-grained estimate of this binding affinity, and that's run through what is called as the SMAX calculation, which is a collaboration with one of our colleagues at London, Peter Coveney, who you might have heard of. And Peter basically said, okay, I'm going to take these top-ranked compounds, run a coarse-grained binding affinity calculation, and then let it go into this latent space representation and try to do something with respect to the learning. And once you do that, we can actually see a lot more points on this landscape highlight with like larger RMSD values and so on. And the DeepDriveMD simulations actually allowed us to probe some of these conformations even more confidently. And we selected some of the top ligands from this data, where we could see, what sort of conformational changes we see with respect to the different effective ligands versus non-effective ligands. And in this case, we happen to see, some of them which are actually like pretty good binders have very different signatures with respect to their binding, because they actually induce a lot of conformational changes and accommodate to the binding site, which then allowed us to reduce the, binding free energy estimates, the uncertainty in the binding free energy estimates by looking at those different numbers. So in some
Excuse
sense,
me, can I ask you, is this binding energy still MMGBSA, or is this free energy?
No, this is free energy based on SMAX, which is a slightly different protocol compared to MMGBSA.
Does it include the energy of the protein?
Yes, it does.
Thank you.
Yes, it does. Yeah, that's also an important thing. I forgot to mention about the MMPBSA difference versus SMAX, but this is SMAX, and thank you for reminding that. And once we do that, we are able to identify a number of these substates and how these binding actually happens. It takes it out of its comfort zone, pushes these compounds and see where it lands. Of course, now coming back to this idea of extensive hardware and so on, we said, if we need to scale it on much larger systems, can we do something more interesting? This is actually a part where one of our colleagues from Cerebras, which is a 15 rack unit system, that's only for training and inference, which is one of the largest wafer scale engine that's ever been built. It's essentially a salad plate of a chip, which people can use for deep learning, training deep learning ideas. And we brought together this heterogeneous hardware and interfaced it with our MD simulations in such a way that we could run MD simulations on our GPU systems. But the training would essentially be run on the CS1, which is this wafer scale engine. And what it allows us to essentially do is we can fold proteins like BBA in under 2 hours using this machine learning type of ideas. And the fact that we can actually process much larger batch sizes, which means that I can actually run very large number of ensembles without having to worry too much about how long it takes to generate the training data. As the training data comes in, I can train and actually interface it as quickly as possible with this simulation. And also look at ways in which we can bind the different proteins. But as you can see from the different iterations, we do reach more or less the sampling of native-like states over the course of these simulations, which was kind of helpful for us because it helped us, show that we can actually get to a point where the protein is folded, it's stable, and so on. So the key thing to remember is the fact that we needed to balance how much of data is getting generated versus how much learning is happening, and then we could run that entirely in a seamless workflow that others could use and so on. Of course, how is this all impacting the SARS CoV-2 therapeutics? I already told you about the 1,000 compounds that we were able to order, of which 50 of them actually show viral inhibition at scale. Several compounds are already being processed for crystallography. I showed you one example of that. We have receptor-specific assays that are also being conducted across multiple labs, synthetic chemistry efforts that are being driven across the different labs again, as well as AI-driven antibody design, which I didn't have any time to cover, but that's an area of interest that we are currently working on. And in terms of the scale of operation, if you really see, we did about 10 power 11 docking calculations using OpenEye and AutoDock with a total ratio of 10:1. Thousands of DeepDriveMD calculations over multiple platforms, a total of 50,000 binding free energy calculations across the different machines, a total of 2.5 million node hours that we utilized, about 25 days on 100% of summit, essentially. This is what it means. And then, we were able to get a crack at least of, 50 compounds that do have antiviral activity, and of which many of them are actually in the process of getting refined further and so on. So with that, I need to acknowledge over 300 people in the team and thank every one of them for all the support that they gave us, including Anda, including a bunch of others that have been helpful. John Stone, especially for all the spike things that we did, helping us through the whole setup and run, and Jim and others for actually supporting what we have done thus far. And of course, the funding acknowledgement to DOE, as well as Argonne and the exascale computing project. With that, I want to thank you all for your attention. And if you have questions, feel free to drop me an email or ask me.
Thank you very much. So I guess this is time for applause, but we're going to skip that or do it electronically. Thank you.
Thank you.
So we have a few questions. I have a couple of questions, but I already asked some questions, so I can save mine for later if you have time. Harold, you had a couple of other questions, please.
Yes, sir. Thank you very, very much. This was a fantastic talk. It's really amazing. It's extraordinarily impressive. I have some specific talks because we have worked along very similar lines at a much smaller scale, of course.
Sure.
So I'm just asking out of deep interest in the approach and in the methodology. What I'm asking is, when you compare the Anton simulations to the deep learning guided simulations, which allow you to eliminate some trajectories and continue with productive trajectories. How do you know what is the and then you show that the deep learning trajectories cover more of the space. What space is that?
Yeah, great question, actually. Thank you. So basically, this is something that there are multiple ways to define that space. And because it's a more general way to actually coordinate what we call as reaction coordinates, this mostly connects with what we think of as the productive end of the space. So if I go back to this slide that I showed you with the folding trajectory, right? I'll go back to that, and I think that's a good illustration for what it constitutes in terms of this training. So if I go back to this folding trajectory, there are multiple ways in which I can actually start from here, 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 anywhere that you say, and then traverse to this folded state, right? And along that space, I'm saying that when I'm in this low dimensional manifold, as long as I'm in a reaction pathway that leads me to the folded part of this landscape, if I'm enriching more of these folded states, then I'm done, right? So the deep learning method is actually picking up on what are those constituent transitions that I care about or that are more important for this folding pathway, and then pushing it along those reaction coordinates, basically.
Understood. So this seems to me to be the equivalent of collective variables, because the folding is a collective variable, namely the fully folded or all native contacts. But some collective variables are more elusive. So saying that you've covered more of the space may have taken you into space that has nothing to do with the space that was covered before. Am I correct?
Yes, you are actually right. So in fact, one of the things that it's a pitfall, according to me, in terms of using some of these machine learning codes carefully, which is that you have to be able to quantitate as to which parts of the landscape you are sampling appropriately, which is biophysically relevant and which are not biophysically relevant.
Exactly.
And I'll give you a experience that we have had with PCA-based methods, where we do see that sort of things where if you just do it in a two-dimensional well, let's say I'm in a two-dimensional well and I'm looking at orthogonal coordinates, I end up in the wrong portion of this landscape because this is unphysical. You clearly see the presence of this high energy states there and you sample them. With the deep learning, one of the things that we have observed, and this is purely empirical, I don't have a way to show that very much in across different simulations and so on, but I'll make that argument in terms of from an empirical perspective. As we were sampling from these trajectories, what we were observing was that it was not going into these invalidated spaces as much compared to PCA based methods and so on. That's the only happiness that we have about this method. There are some aspects of it
I would
which we are
like
not
to have a discussion offline of what are good alternatives.
Please.
If I may, Mr. Chair, can I ask one other question?
Yes, I was going to call you for your second question. Yes, please.
So the manifold that you showed from which you do all the dimensions in the manifold, obviously it's a huge dimensional manifold. Do they all have the same weight? Or do you know which manifold to analyze first and which you should analyze last?
Yes, and I'm going to answer that question a little bit more again in an empirical fashion. So if you're looking at the folding trajectory, you're greedy towards that native state.
Yeah.
Correct? You're greedy towards that native state.
Right.
But let's say my reaction coordinate is about how that ligand is actually diffusing into the binding pocket. Then I have a different sort of a feeling for it. And it's not that these folding pathways and the ligand binding pathways are not necessarily independent of each other. It's just that one is dominant in one case, the other is dominant in the other case. So in that sense, what happens in the case of actually sampling is there is a natural order that you can put based on what we think are essential degrees of freedom for that problem of interest. Especially in the case of ligand binding, this is what we did.
So again,
right?
collective variables?
Collective variables, exactly. Exactly. How do you rank the collective variables is still an open question in my mind. But the one good thing that you can do is you can ask for directions that are relevant for your physics problems of interest. If it is ligand binding, it is one. If it is folding, it's another. If it is something else, it's something else.
That's wonderful. If we have this offline conversation, the solution to this is very similar to what I wanted to propose for the other problems. So we have something to talk about. Thank you very much.
Absolutely. Happy. Thank you.
Thanks. Thank you. So we have a question from Demon Ray.
Sure.
Go ahead and you can ask your question.
Yeah. Thank you for this nice talk. So my question was that is it possible to, when you are talking about the docking of the ligand into the protein in your first part of the talk, so is it possible to include the protein structure as a descriptor so that we can make a model which is transferable between different proteins?
Yes, we have thought about that for sure. There are a couple of reasons why we did not do it for this. One was timing, right? And we could definitely think of ways to express the protein as, feature descriptors and so on. The unfortunate problem with protein structures is that you're always worried about alternate conformations of the different states, of the backbone as well as the side chain and what sort of things can go on. And encoding that information is a little bit more involved. So you need sort of a probabilistic feature description that tells you the transition when you add this particular chemical group here, it varies a little bit by like this or that and so on. Capturing that has always been a hard thing for us, but in chemistry, there is ways to describe that. And we are trying to explore some features from there that we could, exploit for our benefit. But it's been difficult to do it with, just this ones because my group, which is more interested in disordered proteins and how they interact and what they do and so on, getting a continuous description of these is very hard. Has been very hard, at least for us. But open to suggestions and see how that can actually take place. We have I think there are good points where we can actually think of ways to build those accurately.
So currently your method is only we have to train separately for different proteins, right?
No, you generate the training data for different proteins. You generate the training data for different proteins. After all, The docking program is dependent on the protein structure, right? So if you're going to put it, you're going to train it anyway. You're going to get training data. The training as such could be done in one shot, essentially across all the proteins if you want. I would be cautious how to interpret those results a little bit more carefully. We have not done that in our case, but we had the ability to generate the data, so we went and generated it anyways.
So actually, a related question I was going to ask is later, but do you have to try different parameters for your AI model, the number of layers and all the other parameters also, or is it you just do it once and you call it a day and you use it all the time for different proteins and for different days?
So I'm going to be a little bit brutally honest about what hyperparameter optimization, this is what we refer to formally. One of the problems that we observe, and this has been a concern for me for a while, especially with TensorFlow or PyTorch and any of these models, is that depending on your initialization, depending on your, how you instantiate your hyperparameters, you can really be in very weird spaces where you still get good performance on your machine learning task, but the model is extremely unreliable. Right? Make small changes to your input and your results will be drastically different. And while it is an accepted practice in machine learning to do this, from a machine learning research perspective, we have been trying really hard to overcome those challenges, what we call resilience building into our network parameters and so on. So we have a toolkit which is called Hyperspace, which allows you to slightly be better than other software toolkits in terms of hyperparameter optimization. And we have shown that it can successfully scale and do nice things, but unfortunately, it's not that straightforward if you want to be resilient to input perturbations and so on. So that's a very important aspect, something that keeps me up at night also sometimes, because these models are not that reliable, unfortunately. But this is the state of affairs today.
Yeah, fair enough, fair enough. Okay, Shashank and then John.
Yeah, I think you partially answered my question, but just for the sake of clarity, like for myself, so what I was curious about, like when you show these manifolds and you talk about like if we move from blue to red or red to blue, we can do like folding, unfolding, like we are moving from one state to another state. So what if we move along this particular line and take those points as a reaction coordinate to drive our simulations? So can we do that?
Oh yeah. That's actually part of the story of DeepDriveMD, which is that you're taking these, if you project them along those reaction coordinates and start selecting these points as being interesting, you could start off simulations from there and see what happens.
I see. Okay, thank you.
I see. John?
Yeah, Arvind, I just wanted to ask, in terms of the hardware, of course, I'm always a big fan of making new simulation methodologies available to the everyman. And, I was curious in your example about protein folding, being able to do that in a couple of hours on a Cerebras CS1, that's pretty rarefied hardware at this point. And maybe even in 5 years, it'll still be pretty rare to have wafer, the costs of fabricating wafers, single, architecture wafers is massive, and that'll remain something of a unique facility, that you might only find in a handful of centers across the entire country, is maybe even rarer than microscopes, right? And so what I was wondering is, what is the propensity, what's right now the delta between what you can do with the biggest GPU platform you have in terms of performance, or maybe in terms of the scale of the problem? How much of a difference is that CS1 making today? And what do you think the outlook is? I mean, there are people looking at things like die stacking to put memory systems. A large part of what makes the CS1 such a monster is it's a huge memory system on a big wafer. And there are people looking at, could you do something with die stacking with the GPU, or something like a more conventional piece of hardware? But that's probably still, at least 5 years maybe 10 years before we'd have something like that in commodity hardware.
Correct. So
I mean,
I think
just a rough, just a back of the envelope. I'm just curious what the limitations are for the everyman today, and how far, you know, if we assume that a commodity machine maybe won't completely close the gap with the CS1, it gets part of the way there. Where's the crossover where you can do some of these exciting problems, though?
Yeah, I think as you've seen from the spike protein simulations and so on, right? The moment you are at one spike, you're okay with your commodity GPU cluster. Even across 2 GPUs, we are able to roughly train a model and so on and so forth. It depends on how much you want to sacrifice your sampling with respect to the amount of resources available for training, right? So obviously we all know from StatMech and things like that, that more number of ensembles you start simulating, the better your coverage of this landscape is going to look like. And if that is the case, then you want to have as many nodes as available for your sampling portion. And if you sacrifice a big portion, so in my case with the Theta GPU systems, it's only 168 GPUs. If I end up sampling only on 120 GPUs, and I dedicate the rest for my learning, which is still a chunky portion on this one, I still can get some speed up in terms of sampling, but that's not as much because now I'm sacrificing some of these for my, the training part. On the other hand, if I were to offset that by actually using the GPUs, the Cerebras hardware to train and do the inference and all these crappy things, then of the 168 nodes, of the 168 GPUs, I can let all the 168 actually run their simulations. That actually adds on to, how much speed up we get. It's, in some sense, I would say it's not day and night difference per se, but it's a minor but very significant difference in terms of how fast you attempt at doing that. The reason for going with Cerebras in the first place was actually the convenience in some sense, because we knew the hardware from the beginning and so on. And in terms of how popular this is going to be with die stacking and things like that, I think many of the APIs that we are currently using for building these systems. So AMD just released the Daji library and whatnot, and so all these different things that are going on. They are not in a place where we just need to be in terms of getting to where we want to go. Especially if I want to simulate like what Rami says, aerosol effects on the virus and other very ambitious plans that people have had over the last few months. It's been a very tough challenge for us in terms of thinking of ways to do it on conventional machines. And if I want to load this model up on Summit, it's not going to happen. Right? Just
sure,
the practicality of this. That's actually the problem I feel. And I think as more and more hardware companies come along, and there is this chip revolution that's happening, a mini chip revolution that's happening, I think there'll be a lot more fun developments, I feel, where these hardware vendors will actually start interfacing a lot more with traditional HPC realms, because I think people are mostly looking at the cloud as their market now. And I think there is going to be a significant bridge between the cloud and supercomputing resources. And if that were to happen, things would actually change a lot in terms of how the integration would happen. So this is like a baby step example that way, in my opinion, to kind of show if that is possible.
It's very interesting. Maybe one tiny follow-up question. For your cases, which is more important, pure flops, or whether they be half precision or less, or is it memory capacity?
In case of large systems like the virion or the virus simulations that we are doing, I think it's the memory that makes a lot more sense. For smaller systems and things like that, pure flops are helpful. And the reason for it is very simple, which is that you need to sample as much as possible if I'm putting 2 proteins together, or if I'm folding 2 proteins together and so on. I really care about that.
Thank you.
So Payam, did you have a question? I saw you raise your hand.
Yes, hello, thank you for your presentation. I was just a bit curious about the image processing part. How did you define the different atoms, and how did you distinguish between the bonds and atoms? And also, did you use the procedure like the MNIST data list, the data sets, the right-handing numbers? Thank you for your...
Oh, wow. Okay. Quite a few details there. Okay. In terms of the atoms and things like that, it's basically similar to what MD analysis or any of the representations do on the standard processing, right? You're not doing anything this one. And again, on these systems that I showed you, most of these were at the resolution of the C alphas, right? But a few, we have also tried it with the side chains and all those present and so on. We know what we are after. And so the bond definitions and everything is pretty much the same standard that people use in chemistry. In a similar way, there are things that I didn't show here about using graph neural networks and other things that are also present. And those formats are also equally, well defined, and people have paid a lot of attention to how to define them and so on. So, basically, we are standing on the shoulders of giants in terms of that aspect. In terms of how we are. Sorry, the second question was about the image processing, right?
Yes.
Yeah. So that angle was mostly based on just standard definition of how these contact maps or how the point cloud representations are built. It is very similar to what image people do, except that we encode the sequence explicitly, so we know exactly where the sequence is lying. Right? So there is an order in the sequence. We just consider the same order of the sequence from N terminal to C terminus and so on. And of course, and others could comment a lot on the aspects of, box jumping, periodic box boundary conditions and all those things. There is those issues for sure. But when you're doing it in the contact matrix representation, generally you see that the protein stays together. And so you don't run into the sort of issues with the whole system and so on. So we tend to keep them in the same box, and we are happy with that.
We're gonna ask Anda to give us another more detailed presentation on how the workflow works actually. All right, actually it is almost 20 minutes after 4.
Oh, sorry.
So this is obviously a great example of how much and how advanced application of machine learning and AI in molecular systems has become actually, and there is huge room to grow even further, I can imagine. Thank you so much, Arvind, for the very nice and stimulating presentation. And thank you all for joining.
Thank you so much. And if you have any questions, please do drop by an email. And one note of self-promotion in some sense, it's not that we need to do it, but there are some open postdoc positions in the lab. And if you're interested in applying, please do apply. I can help you point the location, because these applications are very different from what I've shown here. This is one part of my bread and butter. There's another part of my bread and butter which I've missed, but that's on the other side of molecular data sets. And so if you're interested, just please contact me directly.
Excellent.
Right? Thank you. Thank you so much.
Great to see you, Harold.
Good to see you. Thank you very much for organizing this, Imhan.
Of course. It's a pleasure.
And thank you, Arvind.
Bye-bye.
Thank you so much. I appreciate it. See you. Bye.